Hi 1001, welcome to week 6 of lab. So last week you had submitted your experimental design proposal. We gave you feedback on that experimental design, gave you some tips on what to improve, how you could earn back some points. So hopefully you've resubmitted your experimental design to get back those points, and we'll give you a little bit more feedback on that. But at this point, this week, what you're going to do is actually start your experiment. So you have a design in hand, you have tasks that you've allocated to everyone in your group, and now you're going to go forward and actually carry that experiment out. So this means that you will be plating up plates of yeast. You'll be manipulating those plates either by applying some kind of UV protective product or manipulating the conditions that they grow in. Whatever your group ultimately decided to test, this is the week that you're actually going to test it. So you'll want to be very careful when you do these experiments to practice good lab safety techniques. More importantly, you want to be sure that you practice good lab sterilization techniques, because these plates can get contaminated, and contaminated plates do not give you any data. So make sure you take your time to sterilize your surface before you actually get going. Make sure you read ahead and understand what is going to be required of you this week, how much time it's going to take, when you're going to do your exposures to UV light, and all of that stuff that is described in the lab manual. Take the time to to plan out your week, and don't get caught flat-footed at the last minute trying to carry out these experiments. Otherwise, it won't work. So that's it for this week. You're doing the experimental work. What you'll be submitting is a picture of your experiment so that we can verify that you've actually done it. And that's it. If you have any questions about this, please don't hesitate to reach out to me or Tom. We're happy to help explain things that are in the lab manual if you're confused. You don't have the virtue of having an in-lab TA, so don't hesitate to take advantage of our time in contacting us for any clarifying questions, okay? That's what we're here for. We want to help you. We want to make sure it works. So don't get ahead of it. Make sure you have a plan for the week, and make sure that anything that's unclear about that plan to reach out and try to clarify with either us or your team, okay? All right, good luck. Have fun. This is a fun experiment. You'll actually be growing some cool microbes and seeing if you actually get some cool results. So we'll see what you get.